Okay, brief update on the mastering AMP situation. I see that most of you got yourselves logged in and got settled with that first assignment, so that's great. Those of you who didn't are still waiting on access code because you purchased a used textbook or something. I am still trying to fight the good fight for you. Pearson did get rid of the access code only option over the summer without telling us. I am trying to summon my inner Karen for you, but we'll see if that works. Never in my life have I asked for special favors for a discount to have someone resolve a problem, but we'll see, maybe they'll do it. Probably peanuts to them. So stay tuned, but for now that 2-week temporary access should still work. So that should get us settled through the next couple of days for sure. So go ahead and take a look at this question. So today we're going to be talking about water, and hopefully we'll also be talking about homeostasis. So hopefully got through the mastering AMP. The answer to this question is pretty obvious, but I want to give you some context for what we're talking about today. A shocking amount of things we care about in bodies has to do with where the water is in your body, how much of it is there, and affects how your heart functions. It affects how your body gets all its nutrients. It affects how your kidneys work. So we spend a lot of time thinking about different compartments of water in the body. So that's going to be one of our first topics today. But before we get to that, we're going to think a little bit about how we can kind of diagram out a body. Now, as an anatomist, this diagram gives me hives, to be honest, but we are going to take a look at it and make sure you understand, because figures in your book are super important. They can summarize a lot of information at once. So if you can understand a picture or a graph, it can be a lot faster than memorizing a whole bunch of words. Really, memorization does play a role in this class, but it's not really what's going to help you in the future as you go into most of you healthcare fields. If you memorize a bunch of stuff right now, probably 5 years from now, that's wrong. But if you understand it, hopefully it'll stick. So what we have here is a simplified body plan. So the goal of this horrible diagram is to show you relationships between different parts of your body. The wrong shape on purpose to kind of highlight these relationships, missing lots of different parts. You'll notice it doesn't have different cell types in here. But what it does show you really well is the external and internal environment. And in fact, on this diagram, they are color coded. So anything here in this bluish, purplish, grayish color is considered external to the body, and anything in yellow or red or orange is considered an internal environment. So the separation here is through something called the epithelial membrane. So that's the barrier. The most obvious one is your skin. So that's what we see lining the outside. So you have that image in your slide. So just because it's kind of clunky looking, I want to make sure that we're all on the same page. So take a second and look in your notes and make sure you can sort of see where the lungs are, where the GI tract or the digestive tract is, find the kidneys and the heart. Let's take a second to look at that diagram. So as you're finding these, you'll notice that while these are all things we think of as organs, for the purposes of determining internal versus external to the body, some of these are internal and some of them are external. For example, if you eat an enormous burrito, like 5-pound burrito, we don't think of your internal self as suddenly 5 pounds heavier. We think of that as moving through a tract, sort of external to your body, until you absorb nutrients, absorb things from that food you've eaten. Okay. So I'm going to draw this out as we kind of go through to highlight different pieces. So when we're talking about the external environment, we're thinking about anything outside the epithelium, so outside this barrier layer. So the most obvious one is the skin. So we have our skin layer. We also have an epithelial lining in the lungs. So as you breathe air in, it's not really inside your body in a physiological sense yet. I don't know why I drew that lung that way. Horrible lung, okay. And we also consider food in the stomach and in the digestive tract as external to the body. Or here we can have some more body on the other side. This is also true of urine in the bladder. We might also consider going out here, part of the external environment. So anything not circled here or in here is external to the body in a physiological sense. So then when we think about the internal environment, we're thinking about what is actually touching cells in your body. So on that diagram, you have just sort of a little clump of tissues. So I'll pull them out and we'll do pink. Okay. So we have just a sort of stylized diagram of random cells in the body for the purposes of this diagram. Our tissue cells. And then we saw a separate segment showing us the circulatory system. So when you looked at the heart, you found something like heart. And then our circulatory system. So the one special type of cell we saw on that diagram was our blood cells. Our blood cells are going to be separately found here in the circulatory system. And we think of them specially because we think about the fluid surrounding those blood cells as being potentially quite different than the fluid sitting around the cells, say, in your knee, right? Because the fluid around those blood cells is blood. So that's blood plasma surrounding those cells. And that may or may not have the same concentrations of things as some fluid out over here. Okay. So as you think about this in the future, you may be thinking about, say, you give someone an IV. What are the different concentrations of different things in that IV fluid that you're giving? How does that compare to the concentrations in the plasma in the blood? Then how does that compare to the fluid surrounding cells elsewhere in the body? And what does that all mean for what's going to go into the body versus out of the body? So that's kind of why we want to be able to discuss all these different types of fluids separately, because they won't always have the same contents, and those contents are going to control how fluid moves. So that can control, for example, swelling or dehydration, things like that. So that's why we have this diagram. Now, as I just implied, there is exchange between our fluid compartments, and there is exchange between the internal environment and the external environment. So things are going to move into your body from the outside, right? So we can follow the air, for example, from your external environment, crossing an epithelial layer, actually crossing a second epithelial layer to get into your bloodstream, right? So gases are crossing here. Or we have nutrients crossing from your digestive tract, again, through a couple layers to get into your bloodstream to be distributed around. We have different things crossing from the blood into tissue, or things crossing from the blood into the external environment, so through your kidneys and then into the bladder, potentially to be excreted as urine. So as we go through this course and we go through the different organ systems, we are going to be thinking a lot about what is moving from one place to the other. What is moving from the external environment? Where is it going and why? Okay, so you don't need to focus on memorizing these terms right now, but they'll become quite important to us, especially in A&P2 as we start to talk about kidneys and digestion. So these are just some terms for different types of movement, different directions of movement. So that's part of why we were highlighting quote unquote relationships with this diagram. This is showing us that absorption is movement from the external environment into the body. That's kind of a normal-ish English word. Secretion, the opposite direction, internal, external. Then we have filtration and reabsorption, which are a little more specific terms when we talk about physiology. So there we have blood into the kidneys, or from that fluid in the kidneys back into the body. But when we get to all those things, we'll dive in quite deep on those terms. So don't focus too much right now, but we'll come back to drawings like this. So we need nutrients and oxygen to get into our body. We need wastes and stuff we just don't need to leave our body. You eat a piece of tin foil from the wrapper of your burrito, you don't need that in your body, it can go out. So we have areas of exchange that we'll focus on. So the major ones between the blood and the external environment are going to be the lungs, as we have gas exchange coming into your bloodstream ultimately. We'll cover lungs in A&P2, same for gastrointestinal tract. We're covering how we get in nutrients from your food. We will see a little bit about that as we talk about blood concentrations, especially sugars in A&P1. Then your kidneys are filtering the contents of the blood really and maintaining concentrations of different ions and such in the fluids in your body. So they have a lot of exchange between the blood and the external environment. So there are different words for the mechanisms of this transport. So in the GI system, we talk about absorption into the body or secretion, or squeezing things out of the body to be excreted. And in the kidneys, we have specific terms called filtration, which is moving stuff from your bloodstream into the kidney. Reabsorption, when we say, oh wait, we didn't actually mean to filter all of that out, and we pull it back from the fluid we put into the kidneys. And we see secretion in the kidneys again, as well as we expel more things into the waste. So for now, one of the things we do really want to get down pat is the difference between internal and external. Okay. So don't have this up as a poll right now, but if you read through these, go ahead and make yourself a little checklist. Which of these are going to be part of the internal environment versus which would be part of the external environment? All right, so the lumen of the lungs, internal or external from a physiological perspective? Who says internal? Awesome. You're all correct then. Lungs, inside your lungs from physiology, external. All right, the whole word right now. About the GI system, who thinks that one's internal? Doing so great. Yeah, within the lumen of the GI tract, still external. About the kidneys, is that internal? No, also not internal. Okay, so these things that you might think about from a day-to-day perspective as being organ systems and therefore inside your roommate, from physiology perspective, these are all still external. They're still part of our external environment. But what is part of our internal environment is going to be the extracellular fluid. So this is going to be one of our fluid types that we are going to want to make sure we remember from this lesson. So our extracellular fluid surrounds the cells inside our body. So we don't want to get tripped up by this beginning extra. So extra does mean outside, but we want to break down this whole word when we're reading it. So it's outside, extra, the cells, but not outside the body, extracellular, outside cells. And I like to break down terms as we go through physiology and anatomy as much as possible, because like I said, I try to memorize as little as possible. There's a lot of stuff to memorize in biology and in healthcare fields. So if there's anything that you can understand just by reading words, we're going to try to do that. So getting to our fluid compartments. So when we talk about all the water in the body, so all the body water, it's just called total body water, TBW. So this is highlighted already in this image in blue here. So note it's not including the external environment, but it's including all the water in the body. Within total body water, we are going to split that out into compartments, into different types of fluid, because we may be comparing different contents of these different parts of fluid. We know that these compartments are going to be separated by membranes inside the body as well. So we looked at the epithelial membrane separating the internal environment from the external environment, but there are also membranes within the body, which if you think about a blood vessel, right, should kind of immediately, intuitively make sense, right? Your blood isn't just sort of gooshing everywhere in your body. It has a container, right? So we're going to have compartments separated by epithelial membranes within the body as well. In this unit, we are going to talk a little bit more in detail about membranes. So try to jog your memories from your prerequisites, because as we're going towards the goal of understanding what is moving across the wall of an organ and why, and is that a problem, we need to be able to remember what can cross cell membranes in the first place. So stay tuned. We'll do a quick review of that. But hopefully you vaguely at least remember this term semi-permeable. The cell membranes are semi-permeable. Semi meaning like some, partially. Permeable meaning things can go across. So some things are going to be able to cross our membranes. Other things are not going to be able to cross our membranes. And that's going to make a difference to movement of ions, nutrients, these various things that we're going to be tracking throughout this course. All right. So as we break down that total body water, we want to be aware that it consists of water, but also things dissolved in that water. So things we're going to pay special attention to in this course, especially are ions like sodium ion. We're also going to think a lot about glucose. So we're going to think about sugar. So there is stuff in this water. So we're thinking about the solutions within the cells. We're thinking about solutions in the fluids surrounding the cells, all of that as we think about the total body water. So as we start to get more specific, now we don't want to know just about like all the water in the body. We want to start to talk about specific locations. We're going to think about compartments. We're going to think about those specific fluid locations. So the first division that we want to pay attention to is whether the water we're looking at is inside a cell versus outside a cell. So going back to that warm-up question at the beginning, that is the first way that physiologists are going to divide all that water in the body. First thing we want to know is it inside a cell or is it outside of a cell? That matters to us. So if we take total body water, we can make kind of a flow chart. Right? So we're going to first separate into intracellular fluid. So intracellular fluid, intra means inside, cellular referring to the cell. So inside the cell, and fluid because it's water, but also these various things that are dissolved in the water. And our other main division is going to be just the opposite, right? If we have the inside the cell, we also need the outside the cell. So coming back to that extracellular fluid, anything outside the cells, so long as it's still inside the body, is ECF, so extracellular fluid. Any questions to this point? So this should be a great pause point in your minds, but we are going to keep going. Okay. So we have our intracellular fluid compartment, and that one's relatively simple. So we have our total body water divided. Sorry, I'm going to keep drawing that little flow chart because it's helpful to me. Okay, so we have our intracellular fluid, and we have our extracellular fluid. So with our intracellular fluid, we don't need to think any further. If it's in a cell, it's in a cell. That's it. So when we think about the fluid inside the cells, really what we're thinking about is the cytoplasm, right? So that internal sort of matrix of the cell where we have all our organelles floating around, we have whatever sort of metabolic byproducts are in that cell. They're all just kind of inside the cell. So we don't have to think any more about intracellular fluid. We don't have to think, is this a kidney cell? We don't have to think, is this a cell in a muscle in your leg? Just in the tissue generally, that's ICF. The ECF is a little more complicated. The ECF we are going to break down again, which is why I was just drawing this little flow chart. So the extracellular fluid is everything outside of a cell. But here we are going to be a little more specific because we want to know, now, is it just generally somewhere in the body outside the cell? Or specifically, is it in the blood, but not within like a red blood cell? So our next division, I should have drawn this a little farther up. All right. So we're going to divide after we divide that total body water into internal and external, we're going to divide the external again. So the extracellular fluid now gets divided into specifically, are we talking about interstitial fluid, which is just surrounding general tissue cells? So that's our ISF, interstitial fluid. Not sure I remember what the root stitial means, maybe space. Anybody good at Greek and Latin roots? Okay, well, just remember it's an S instead of a C. So our other division of that ECF is going to be the plasma, because plasma has different concentrations of things than the ISF might, especially if you were to give someone fluids, for example, if you were to hook them up to an IV, put some type of solution into their bloodstream, right? Then we'd have a really big difference between fluid just surrounding tissue cells generally versus what's now in our bloodstream. So that's why we divide these two things separately. So we think about this extracellular fluid as the internal environment for any given cell. Because if you think about it from the perspective of, say, this tissue cell here, right? If it looks outside itself, right, what it's looking at, right, it just sees its neighbors. What it really sees is just that interstitial fluid surrounding it, right? That's its immediate external environment touching the outside of its membrane. You are in the bloodstream and you're a red blood cell. Your immediate environment, right, if you look around you, is the plasma, right? So any one of these little cells doesn't have to think about, is there a tree or something over here outside the body? That doesn't matter to a cell inside your body. So we think of that as our environment, our internal environment for our cells here. So here we start to get to how much of this water is there, how much of this fluid, I should say, is there. So total body water, so all the water in an average body has about 42 liters. So that's our typical volume. So whenever you're looking at like an anatomy or physiology textbook, I should say that when they give you amounts in liters rather than like percentages, they're basing that on a 70 kilogram male person. So any individual person is going to vary from this for all sorts of different reasons, which will hopefully really be emphasized to you in the anatomy lab when you see our 4 donor bodies and you look at that individual variation. So this is kind of like a physics problem kind of person, right? This is not a real person who exists. It's just a sort of generic average. Okay. So about 42 liters, 60% of body weight is our water inside the body, that total body water. Now we divide it between the ICF and the ECF. So that's our first division. Is it inside a cell or is it outside a cell? So we have our 28 liters versus our 14 liters here. So we add those together and got our total body water again. And then we divide down the ECF further between, is it in the plasma, which you have about 3 liters of fluid in your blood, versus is it in the interstitial fluid, so just sort of generally surrounding cells, about 11 liters surrounding the cell. So that breaks down to about 60% of body weight, kind of like 50 to 60, as being our total body water. Then of that, we divide 40% of your body weight is intracellular fluid, so the cytoplasm basically, versus 20% of body weight, that body fluid, is the extracellular fluid. And then we're breaking down the plasma versus the interstitial fluid. So we covered these questions. So throughout your slides, various checkpoint questions will be presented. Sometimes we'll talk about these in class. Sometimes we'll use them as warm-ups or closing questions, but otherwise you should be sort of going through them as you're reviewing your notes, and make sure that now that we've covered material, you've consulted your textbook, that you understand these questions and are able to answer them. And if you can't or don't understand the questions, ask the next day in class, come to my office hours. These should be things that you should be able to explain to yourself as you're studying and preparing for the end of the unit. So a huge topic in biology, but especially in anatomy and physiology, is understanding how we have balance, right? So your body functions at a specific temperature, functions at a specific pH. You want the same amount of blood in your body day after day after day. You want to keep the sort of surrounding of any given cell consistent day after day after day, so that that cell can continue to function. If you suddenly heat a cell way up to 120 degrees, right, it's not going to function the same as at normal body temperature. That's a problem. So a lot of the mechanisms we're going to talk about are how your body adjusts when something happens, when there's some kind of a change. How does your body compensate for that? How does your body get back to that balanced state where everything is predictable for all your different organs? So this is our concept of homeostasis. So that's our second lecture objective for today. So when we think about homeostasis, what we're trying to do is maintain a consistent internal environment. Now, there may be some fluctuations. We do think of it as sort of like a dynamic balance, right? But we're trying to stay basically a sort of regulated set point. Now, not everything in your body is regulated, just the really important things, the things that would seriously, detrimentally affect your cells, affect your organs, things that would cause serious, serious problems for how your body works. So some things that are going to be regulated through homeostatic mechanisms, bless you. Temperature, your body temperature is really important, which you all know, right? If you're feeling sick, get a thermometer, take your temperature, see if you have a fever. It's part of how you tell, is there a problem, right? So on a normal day, your temperature should be remaining relatively consistent. When we say we're regulating volume as part of our internal environment, we're really talking about is like volume of blood, right? It's a big problem for your body if you suddenly have way less blood, right? Like if you're bleeding out, that's a problem. We're maintaining the volume. When we say composition, again, we are often thinking about the composition of blood or the composition of the other fluids we're thinking about. So potentially the composition of the interstitial fluid, the composition of the internal environment of a cell and cytoplasm. So we're thinking about the concentration of, say, sodium outside a cell versus inside a cell. It's actually going to be really important in our next unit when we think about how nerves work, right? So if we alter the composition of these fluids, we alter what crosses a cell in which direction, which is going to alter fundamentally how some of your organ systems work. Now, homeostasis, probably a word you've heard a lot before in the past, right? I can't imagine a biology class that doesn't say the word homeostasis, but maybe there are some. But it's actually a pretty complex process. We're going to be thinking about things like feedback loops. We're going to be thinking about multiple different organ systems involved. We're going to be thinking about all types of sensors. This is really important to your body. And there are a lot of parts of your body that are just focused on maintaining this consistent internal environment. So the main purpose of a lot of our physiological mechanisms is to maintain homeostasis. If we disrupt homeostasis, if we disrupt this balance, if we suddenly have a temperature of 110, this is the basis for disease and death, right? If we seriously mess with these regulatory mechanisms, we can no longer regulate these properties, you're going to get really sick. So we need to pay attention to these feedback loops and understand how these organ systems are going to work together to maintain homeostasis. And that's a big focus of our course. So when we're thinking about feedback, when we're thinking about homeostasis, we're going to be using this word feedback a lot. Okay? So we'll have negative and sometimes positive feedback. And in the future, as we get into different organ systems, they'll be kind of like marked on various diagrams, like, oh, this is a negative feedback loop. So right now we want to understand what that is. But in very simple terms, right? If you're suddenly start running down the hallway, someone yells at you, stop running, negative feedback, and stop running, you go back to walking. Your body has basically the same kinds of loops, right? You notice a change, it's not good. There's negative feedback that sends you back to that balanced state. So we're going to go through a lot of sort of more complicated processes about exactly how that happens. But this term actually does, at its core, kind of mean the same thing it would mean to you in your daily life. But we're going to graph it a little bit. So when we're thinking about negative feedback, you'll see graphs something like this. So usually on the x-axis, what we're looking at is time, right? So we're looking at changes over time, right? And this is just going to be the amount of something. This is whatever variable we're looking at on the y-axis. So I'm going to leave that blank for now. What we're looking at, so we're going to start with some type of a set point we're trying to maintain. Our set point. So you go out, you take someone's temperature every day, right? We're trying to keep, say, 98.6, right, as our set point, right? But if we notice it spikes up, right, we notice suddenly they have a fever. Now we have an error signal. So that error signal here is that deviation from our set point. So our error signal is how different. How much higher did the temperature get there? Right. So hopefully our body will adjust this fluctuation in temperature. If it doesn't, we might do something like give them some Tylenol, give them some kind of fever reducer to bring their temperature back down towards the set point. So that would be considered negative feedback when we have a change, right? It went up and then we pull it back down, right? So negative feedback is whenever we have a change in one direction and then a mechanism that pulls us back towards the set point. When we have a change up, right, a temperature gets higher, that's a little easier to understand. Occasionally people get tripped up. The opposite is also true, right? If you're suddenly really, really cold, say hypothermia is setting in, right, and your body temperature is dropping like crazy, we still have an error signal in this downward direction. And we still want to change that body temperature, but this time we're trying to pull it back up. We still consider this negative feedback because it's an opposite. So the negative here is really referring to do the opposite thing, not so much does this number need to go up or down. So we'll look at positive feedback in a little bit, but positive feedback, a graph of positive feedback would look something like this. Just like asymptotic, straight up and up and up. So that's kind of a snowball situation where something in your body changes and then your body says, yes, do more of that. And you do more and more and more and more. So that's going to be our difference between negative feedback and positive feedback. We are almost always talking about negative feedback in this class, but we'll see some cases in our reproductive unit where we have positive feedback. And we'll talk about positive feedback when we talk about blood clotting as well. Two big examples where we see this other rarer type. So we can also think about this in sort of daily kind of experiences that we have. We can compare this to, say, a car set at cruise control, right? So you have a speed you're trying to maintain. as you go up a hill, right? You're driving somewhere, going up one of these big steep hills we have in Duluth. Naturally, if you didn't have cruise control on, right, your car would start to slow down because gravity's pulling it backwards. So you'd either have to hit the gas pedal or let your car hit the gas on its own to get back up to that 60-mile-per-hour speed. Our body's doing the same thing, right? So we have our constant set point. We notice an error signal as the concentration of something starts to change, the temperature starts to change, your blood volume starts to change. And then we're going to have some type of mechanism that pulls us back up to the set point. So the set point is that specific number we're trying to keep under control. So on this car example, say 60 miles per hour, or in terms of your body temperature, say like 98.6. Those are our set points. We should talk in metric in this class. So core body temperature is supposed to be 37 degrees Celsius. Some compositional things, your sugar levels, blood glucose should be at 100 milligrams per deciliter. Your blood pH is at 7.4. These are all examples of things that are regulated variables in the body. So we'll talk about them at various points throughout this course. So the regulated variable isn't the specific number. The regulated variable is just temperature, sugar, pH generally, and then the set point is the value. Questions right now? Great. So if a regulated variable changes, then in negative feedback, we're going to have the opposite change. So here, our example, right, is if our variable decreases, then the system is going to respond to make it increase. Or in the diagram here, blood glucose rises, so we have an increase, and we ultimately then need a decrease to get back to that set point. It's really just, we're trying to self-correct, get back to that original value by pulling whatever it is in the opposite direction. The error signal is just that deviation, that difference between where we want to be and what we've noticed, right? So what our sensors have sensed, whether it's a temperature change, a pH change, whatever. In order to do this, we're going to have different organ systems, different parts of the body involved. So we can kind of generalize, and you will see sometimes in the figures we see in the future, this color system may be consistent. So make sure to read keys here. The things that we're going to be paying attention to when we think about homeostasis are sensors, right? So how do we know about this change, right? You may know that someone's temperature changed because you gave them a thermometer and took their temperature, but how does your body know, right? We need a sensor. We're also going to need somewhere in the body to process that information, right? So when we have a sensor in the body, usually it's just really just sensing that one thing. If you think about like chemical composition, maybe that makes sense, right? Like you have a receptor, it's like, oh, there's a lot of this ion, right? But a receptor just somewhere in your neck can't do anything about that. It has to communicate that information somewhere. The integrating centers in your brain are largely where that information is going, so that we can create a response in the body to adjust this change. Okay, so that's our integrating center. We're sensing the change, going to the integrating center, and then our effectors are what has an actual effect on the variable. So here we're looking at blood pressure. So they're saying the heart rate is going to affect the blood, right? So what actually does something is your effector. I'm going to go through and trace out this full example here. So, you know, if you've ever had this experience where you're sitting or you're lying down and you stand up and suddenly you get really woozy, maybe your vision gets kind of like blackish and has stars. If you have low blood pressure, this may particularly happen. So this is because if you're lying down, your blood is all kind of like flat, right? You have a hole in your blood down here, down here, down here. When you suddenly stand up, all your blood is affected by gravity, just like everything else in the physical world. So suddenly it gets pulled down towards your legs. If you have a high enough blood pressure, and if your regulatory mechanisms are fast enough, you don't notice this. But under certain conditions, or if you have particularly low blood pressure, you may actively notice this change in blood pressure. So what we've had here is we had our stimulus for the body to respond, which was this falling blood pressure, right? So our stimulus is the falling blood pressure. And we have blood pressure centers throughout the body that, or blood pressure receptors that sense this throughout the body. So you have some in blood vessels near your heart. You do have some in blood vessels in your neck. So they can actually sense blood pressure went down. That's our sensor. So we can think of it as step one. They send that signal to your brain, say, hey, brain, our blood pressure is low. This is a problem. That's our integrating center, step two. And then step three is your brain says, oh, okay, we're going to fix this. So it's going to send signals out to our effector organs, like the heart, but also other blood vessels, also potentially other organ systems, depending on why exactly this problem is occurring. So the heart would be an effector organ here, but we might have other effector organs. It's not the only effector organ. So these are all just kind of categories of types of parts of our body. So here it is written out as definitions, if anyone's a big flashcard fan. So the receptors are going to detect stimuli. So we have thermoreceptors for temperature. Thermo means temperature. Chemoreceptors, heme here for chemical, so like chemical concentrations, things. Baroreceptors, like a barometer means pressure. Okay. Then we have signals that are going to allow different parts of our loop to communicate. So we might have a signal from a receptor, so from a thermoreceptor, one of these temperature receptors, to the integrating center. The integrating center would orchestrate a response, so plan out how we're going to fix this. Then there would be another signal out to the effector organ, which is ultimately going to have a response. And this is a pretty consistent list of effector organs. So when we think effector organs, we are going to see our effector organs are going to be muscles and glands. I can't think of any counterexamples right now. So muscles and glands are effector organs. Okay. So that's it for today. I'll stick around for questions, but we'll pick up here on Monday.